This is the second lecture in our look at the human senses. The first special sense we're going to talk about today is hearing. I'm going to adjust this a little bit so you can see the picture. The structures involved in hearing are actually grouped by where they are located, whether they're outside of the structure of the head, right in the inner cavity, or all the way buried within the temporal bone. So the outermost structures are appropriately called the outer ear. That starts with what we know of as the ear itself, which is actually called the auricle in Greek and the pinna in Latin. And that's just the funnel-shaped structure that's made up of cartilage that kind of collects the sound waves from the air and then shoves them inward toward the actual receptors within the temporal bone. The passageway from the outside of the ear into the inner structure of the ear canal is called the external acoustic meatus. So acoustic is a term that refers to sound. So this is just a connection point, like a hallway for the sound waves. And that hallway routes to what we know of as the eardrum, which is called the tympanic membrane. And that is a very, very tight membrane that's made kind of like a thin layer of skin. And when the sound waves from outside the body hit that eardrum, it vibrates. And then on the back side, on the inside part of the eardrum, it funnels down to a point, which helps to concentrate the sound waves, at which point they are passed to the middle ear. So the middle ear is completely housed within the temporal bone, so the bone of the skull. That's on the inside behind your ear itself. And inside the middle ear are the three tiniest bones in the body, and they are called the malleus, the incus, and the stapes. So malleus means hammer in Greek. Incus means anvil, which is like a kind of rectangular shaped structure. And then stapes means stirrup, like you would see on a saddle, for example. That just refers to the shape of those structures. And so as the eardrum vibrates, it passes the sound waves through each of those bones in turn, and as it moves through each bone, it gets more and more concentrated until eventually it reaches the oval window of the inner ear. Another interesting feature of the inside of our ear region is, actually that color's not really going to work, is this structure right here, which is called the auditory tube. There's one in each ear, and it leads downward to the nasal cavity. So it actually connects what's happening with your nose and mouth to what's happening inside the ears, and it helps to maintain pressure. There's air in there, and there's also a little bit of fluid, and that helps to keep pressure so that the eardrum is stable and it's not kind of pushing outward into the ear canal or collapsing inward toward the inner ear. So the inner ear itself has quite a lot of different structures. We're going to focus on just a couple. So one of the major structures is called the labyrinth, and a labyrinth, oh geez, that was a little more dramatic than I expected it to be. The labyrinth is a complex system of tubes and a bunch of different chambers that have varying volumes and size, and each of those tubes are filled with fluids. The significance of that fluid is that it shifts with waves, just like the ocean or a bathtub or a lake. The fluid shifts and has different kinds of waves, so different sound waves have different impact on the fluids found in the inner ear. There are also some structures in the inner ear that have nothing whatsoever to do with sound and everything to do with balance and equilibrium, which we will talk about in the next slide, but those are called the semicircular canals. So just take a look at how they're positioned, how we have one on the horizontal axis, one on the vertical axis, and one kind of laying on the diagonal axis. The final structure that you should know of the inner ear is the cochlea, which is this little kind of cinnamon roll or snail-shaped structure. It is filled with tiny, tiny hairs and also again with fluid. So as the sound waves ultimately reach the cochlea, that's where the oval window was found, the fluid passes the waves through, and depending on the strength of the waves, the height of the waves, the frequency of the waves, we get different kinds of sounds. So those are picked up by mechanoreceptors. They're actual physical hairs that pick up differences in the way the waves are moving. And then they pass that information to the auditory nerve, which is found right here, to be processed in the part of the brain that deals with sound, which is found in the temporal lobe. Okay, as promised, here we're going to talk about equilibrium. So equilibrium is in terms of senses, is your body's ability to detect change in the environment. So that change might be inside the body or it might be outside of the body. So in order to detect change, we have to kind of be constantly monitoring when things are consistent. That is the idea of static equilibrium. So static means unchanging, and static equilibrium describes how your body detects where you are positioned in space and time. So whether you are positioned in an upright posture, whether you're laying down, whether you're sitting, and it helps to give your brain information about what's happening with your body posture and position relative to the things around you. So the way that happens is using an organ called the vestibule. It's found in the inner ear, right by the semicircular canals and the cochlea. And again, it's filled with fluid. So equilibrium is another sense that is detected using mechanoreceptors. It is a mechanical change in the way that the hairs are situated. When the fluid around the hairs, deep inside the vestibule and the semicircular canals, when those hairs bend and move, that gives your brain information about what's happening. In order for the fluid to be moving, your body must be moving, or at least your head must be moving. Then that information travels along the vestibular nerve and then back to the cerebellum, which is ultimately responsible for equilibrium, posture, a sense called, holy cow, a sense called proprioception. Proprioception, which basically means balance, posture, and awareness in space and time. Dynamic equilibrium is a look at how your body is changing position as it's happening. So if you are running or jumping, or you go from sitting to standing to walking, the fluid within the semicircular canal shifts, and then it gives your brain information and it helps you to stay balanced as you're moving. You don't need a lot of balance most of the time when you're stationary, but as soon as you start moving, you certainly do. When the brain is taking in lots of information, it has to combine that information to the likeliest scenario. So when it feels movement and your eyes see that you are moving, then that tells the brain, I'm in motion. This is what happens with motion sickness. The eyes tell the brain that you're moving when you're riding in a car, but the equilibrium sense is that you're not moving because your body is sitting still. You're in static equilibrium. You're holding your position. And so the brain gets confused because the inner ear is saying, nope, you're still, but the eyes are saying, nope, you're moving. And sometimes the skin is feeling the vibration of the car or the bumps in the road. And so the brain gets confused. And when the brain gets confused, it often responds with lightheadedness, nausea, feelings associated with motion sickness. Okay, our final special sense is vision. So I'm going to suggest that you pause right here and write down all of these structures, and we'll talk about them on the next slide. Okay, let's talk about how those structures that you just wrote down work together to create an image for the brain to process. So first we have to get light rays into the eye. And the way that happens is we have this open structure here called the pupil, right? You just saw that. And some kind of an object, let's say it's a tree, way out here. So as light rays hit the tree, they bounce back and head toward our eye. So they're moving in little waves like this. Those waves travel through the pupil and into the inner part of the eye. We have the ability to control the amount of light that comes into our eye depending on the amount of light that's in the environment. So when we're in situations of very bright sunlight, for example, the pupil muscles tend to contract, which shrinks the pupil size down and lets in less light. If it's a very bright situation, we can still see fine with less overall light in the eye. If it's a very dim situation, say like a movie theater, for example, the pupil will relax and let lots of light in so that we can still pick up objects and information in our surroundings. Another thing that's really critical about the way that light enters the eye is how those light waves move. So as the waves are moving through the air, it's moving through a gas, which is one type of movement. But then as soon as these light rays hit the front of our eye, which is the cornea, that's a solid, so that bends the waves slightly. Then those waves pass through the pupil, through this liquid called the aqueous humor. So now they're bent again because they went from a gas to a solid. Now they're moving through a liquid. Then they hit the lens, which is another solid, so they're bent again. Now they're moving through the vitreous humor, which is another liquid, so they're bent again. And eventually they all kind of converge or meet at this point on the retina if everything is working properly. So we get the rays bent in lots of different ways that ends up focusing them into one tiny little location on the retina. That process of bending is called refraction, and it is critical to our ability to actually form images and to quote unquote see the world. One thing that's interesting is because of the way the rays get bent, it focuses the light really well, but it also flips images upside down. So the image that exists on the back of the retina is actually upside down compared to the world. The receptors that then take in that light information are found all along the back of the retina. And there are different kinds of receptors for different kinds of light. We generally group them into kind of two categories. So rods are the type of cell that collect information on black and white vision. So shadow, detail and shape, things of that nature. And they're very sensitive. So that's how we are often able to see things in very, very low light situations. We can still kind of pick out the rough information of a room, for example, even if your lights aren't on. The cones, however, provide color vision. The way I remember this is cones is CO and color starts with CO. So cones provide color. They also provide more sharp detail in objects. So the rods might tell us that there is a large object on the other side of the room. The cones help us to see that maybe that large object is a couch or a bookshelf or something of that nature. We get more detail and edges. The most highly sensitive portion of the retina is called the fovea centralis. And it is a region that has a huge kind of packed zone of cone-specific photoreceptors. So when we are making an attempt to see an object in as much detail with as much sharp focus as possible, your eye is designed to refocus and to bend the light rays so that the image falls specifically on the fovea centralis. Your eye will adjust in terms of location and focus to do that for you. When information leaves the back of the eye, so here we are, right, looking at our eye, it travels along one of two optic nerves. We have a right optic nerve and a left optic nerve. And then that information comes together. We've seen this image before at the optic chiasm. And then we combine left side visual field information and send that to the right side of the brain. And then we combine right side visual field info goes to the left occipital lobe. Left side visual field info goes to the right occipital lobe. That gives us one mostly accurate, mostly complete 3D picture of the world with as much detail as we can provide. And then the brain kind of fills in all the rest. Anything that's missing or seems odd, the brain adjusts and gives us this picture that seems the most likely for the environment that we're in. Okay, here are your summary questions for this set of notes. Please answer these as much or as well as you can, and be sure to come in with 2 or more discussion questions for class. I'll see you soon.